All right, so today we're diving into one of the biggest, most fundamental challenges in all of genetic engineering. How in the world do you get DNA from one organism into another? It's all about the delivery, and trust me, the solution scientists came up with is absolutely ingenious.
So let's frame the problem. Picture this. You've got the human gene for insulin right here in your hand. And over there, you've got a single bacterium. Your mission, should you choose to accept it, is to get that gene inside the bacterium. Now, you can't just, poke it in there. A living cell has its own security system, and it is not a fan of uninvited guests.
And that, right there, is the heart of the gene delivery problem. A cell isn't just some passive little blob. Nope. It's more like a fortress, and it's got all of these defenses specifically designed to find and destroy exactly the kind of foreign material we're trying to sneak in.
Yeah, the cell's defense system is incredibly good at its job. If you just drop a piece of DNA in there all by itself, these enzymes called nucleases immediately swarm it. They're like molecular shredders, and they will chop that gene into completely useless bits in seconds. So your precious genetic code is gone before it even has a chance to do anything.
So if you can't send the gene in alone, what do? Well, scientists figured out a workaround. You package it. You put it inside a special delivery vehicle, something that the cell will not only accept, but will actually copy for you. And that vehicle is called a vector. At its core, a vector is basically a genetic taxi. It's this little piece of DNA, usually a circular one called a plasmid, that's been engineered to do one thing really well: carry our gene of interest safely past the cell's defenses. It's the ultimate Trojan horse, getting our cargo inside the city walls without raising any alarms.
But, not just any old piece of DNA can be a vector. It needs some very specific features to get the job done. So let's pop the hood on one of these things and see what makes it tick.
Okay, first thing, and this is absolutely non-negotiable, it needs an engine. This is called the origin of replication, or just ORI for short. It's a specific DNA sequence that basically screams at the cell's own machinery, hey, start copying right here. Without it, the vector is a one-and-done delivery. But with it, every single time that bacterium divides, our vector and the insulin gene it's carrying gets copied too. Next up, every good vehicle needs a cargo bay. For a vector, this is called the multiple cloning site. And it's just a brilliantly designed little stretch of DNA that has a whole bunch of different landing spots for what you can think of as molecular scissors. This means scientists can use this same basic vector to carry all sorts of different genes. It's like a universal adapter, making it an incredibly flexible tool. And finally, you need a tracking system. This is the selectable marker. And honestly, this is where the design gets really, really clever. See, the process of getting a vector into a cell, it's not very efficient. Most of the bacteria in your experiment just won't take it up. So how do you find the ones that did? Well, you give the vector a superpower, usually a gene that makes it resistant to an antibiotic. Here's how that works in practice. After you've mixed your vectors with the bacteria, you just dump an antibiotic into the mix. Now, any cell that didn't get the vector is immediately killed off. But the very few that did successfully grab one now have that resistance gene. So they survive and they grow and they multiply. It's a brilliantly simple, if a little ruthless way to filter out all the failures and find your winners. Now, just like you wouldn't use a sports car to haul a bunch of lumber, scientists have a whole garage full of different vectors. The vehicle you pick really depends on the job at hand, and specifically the size of the cargo you need to move. And that toolkit is seriously diverse. For small jobs, like a gene that's maybe 20,000 base pairs long, a simple plasmid is perfect. It's like a motorcycle, fast and agile. But what if you need to move a huge chunk of DNA? Well, then you need the heavy hitters. We're talking about things like bacterial or yeast artificial chromosomes, BACs and YACs.
These are the cargo ships of the genetic world, capable of carrying 1 million base pairs or even more. It's all about scale. So the bottom line is pretty straightforward, right? The mission dictates the vehicle. The bigger the piece of DNA you need to transport, the bigger and more specialized the vector you have to choose from that toolkit. Okay, so beyond just carrying the DNA, vectors can have one of two very different missions once they're inside the cell. It's not just about the delivery itself. It's about what you want to happen after the packages arrive. And this is a really important distinction. The first type is a cloning vector. Its only job is to make more copies of the gene. Think of it like a genetic photocopier. You put one gene in, you get millions back. The second type, an expression vector, is totally different. Its goal is to actually get the cell to read the gene and produce the protein it codes for, like our insulin. It turns that little bacterium into a microscopic factory. To make that factory work, an expression vector needs extra tools, extra instructions. It needs a promoter, which is basically the on switch. It needs clear start and stop signals for the protein-building machinery. And some of the really sophisticated ones even include a purification tag, like a tiny molecular handle that lets scientists easily grab the finished protein and pull it out from all the other stuff in the cell. And this is why all of this really, truly matters. This isn't just some neat trick scientists do in a lab. Vectors are the absolute workhorses that make so much of modern medicine possible. It's how we get bacteria to pump out massive quantities of life-saving insulin. It's how we develop new vaccines. And it's the foundational technology for gene therapy, which aims to deliver correct versions of genes to patients and fix genetic diseases at their very source. So think about that. We've built these incredible genetic vehicles. They're powerful, they're precise, and we can program them to deliver almost any code to almost any cell. The technology is here, and it works. So the only real question left is, where do we tell them to go next?